So now we are on part 3, which is the final installment in this series of lab videos. So just a recap, we put the C. elegans in a high salt environment and they looked dead or dying, but then 48 hours later they seemed happy again. I'm going to just quickly show you that video one more time. Here they are moving around, really happy, as fast as they were before. We talked about there's a lot more of them before. So really now looking at what happened. And you should have also read the glycerol reading and answered the questions in between so that you have a frame of reference and understand kind of what's going on. This is where we left our understanding before, in that there was an environmental stimulus where there was a high salt environment. There was then a biological response where through osmosis the worms got seriously dehydrated. But now we can also understand there was some gene regulation activity. So once that happened and the worms got dehydrated, now one of the things to notice is that the signal in this case was actually the worms getting dehydrated, not the high salt. But don't worry so much about that detail, but that's just something to kind of keep in the back of your head, that the signal was the dehydration, not the salt. And that turned on a gene that regulates the production of glycerolase. If you don't know what glycerolase is, please go back to the reading. And a protein was produced, right? So we've talked about genes lead to the production of protein. So that protein was glycerolase, which catalyzes the production of glycerol from glucose. And again, that's all in the reading. And then the resulting trait was that these worms now have the ability to live in high salt environments by essentially producing a glycerol shell that coats the membrane of their outer cells. So hopefully you were able to kind of get there on your own and put all of the pieces together. Now let's do a little bit of breaking down of what's happening in each cell. So I'm going to have you make 3 C. elegans cell diagrams, one from a C. elegans in a low salt environment, one from a C. elegans recently moved to a high salt environment like 15 to 20 minutes prior, and then one from a C. elegans moved to a high salt environment several days prior. And think about like the lactase cell diagrams that we made in the gene regulation notes, but also if you want to kind of use the first one as an example, the C. elegans in a low salt environment, you're not even sure where to start, then that's fine as well. But then I do want you to try the other two on your own before you look at the answers. So if I'm drawing a C. elegans in a low salt environment, the first thing I need to draw is just the cell with the nucleus in it. Okay, so this is the cell and this is the nucleus. Generally we make them oval because it fits better on the page and you can fit more diagrams on a page. You can make yours more circular if you like. And then I'm going to put a chromosome into the nucleus, and then I've also put on a few lines here and here to say this is the gene that we're looking at. So this is going to be our glycerolase gene. And some people will choose to kind of make like lines to kind of zoom it out of the nucleus. You don't need to do that if you don't want to, but if you want to, and then you can put in a regulatory portion and a coding portion, that's fine too. It's your choice. So I'm going to put the repressor on, and if you go back to the previous slide, you'll see what all of the symbols are. These are the symbols that I'm using, so if you want to copy those down on your sheet so that when I'm going through the example you have them, I would recommend pausing the video and doing that right now. So I have my cell with my repressor protein on the gene. And again, if you want to kind of zoom in on this gene and have a repressor region or like a regulatory region and a coding region, that's totally fine. You can do that. Okay, so then the next thing I'm going to do is I'm going to put in a little bit of salt. Now low salt and high salt is a little relative, and so you'll see on my high salt environments I just have more salt. And then I'm going to put in some glucose. Actually all organisms need glucose, whether they do complete cellular respiration or not. They're still going to use some glucose for energy. And then I'm going to put in a lot of water. Okay, so I've got some water in the cell, I've got some water outside the cell in the environment, and then I've got some water, if you remember, through osmosis. The water is going to be moving in and out through water channels in the cell membrane, but you've got some water moving in, some water moving out, and it keeps a nice equilibrium within the worm. And so this is what the C. elegans in the low salt environment is going to look like. So I really do want you to pause the video and you try to do yours in the high salt environment after 20 minutes and then after about 48 hours. And then I am going to show you my pictures so that you can understand them, but if you're not trying them yourselves, then all you're really doing is kind of copying what I did rather than learning how to build these and work through it yourself. So I really, really encourage you to do that. Or not to do that, depending. So in a high salt environment move recently, say 15-20 minutes ago, the first thing I'm again going to have is my cell with my chromosome with my repressor on the gene. And now I'm going to pause this and plug in my computer. Okay, and so the first thing I'm going to do then is put in more salt. And so you can see I have more salt than I did in my low salt environment. And what's important here goes back to what I said before, that the signal to regulate the gene is the dehydration, not the salt. Because if it were just the salt, then we would expect there to be an activator now. But that can't happen until after the worm has become dehydrated, because it's the dehydration that's actually the signal. I'm going to put my glucose back in, I'm going to put my water back in, but now because there's such a high concentration of salt outside the worm, if you look, you'll notice I've got 3 arrows moving out and only 1 moving in. And so I've got more water flowing out than in, meaning there's a net flow out, which is going to cause my worm to get dehydrated. And that's the signal to activate this gene, that dehydration. So now I'm going to go ahead and show you what's going on in the high salt environment. In the high salt environment after about 48 hours. So again, I have everything I had before, except now I don't have the repressor on there, and we'll put an activator in a little bit. I'm going to put my salt in. I've got both my water and my glucose. This is exactly what I had before. But now I'm going to put an activator on that gene instead of the repressor. And because I now have that activator, now I can make glycerolase. So that's my enzyme glycerolase. And glycerolase is going to change the glucose into glycerol. So the glucose moves in, and then what comes out is the glycerol, and that moves out into the cell. And remember, this is an enzyme, so the same enzyme can be used for multiple molecules of glucose. Now, if you're drawing this, obviously you don't have the benefit of cool PowerPoint animation in your journal. The way I've always drawn this is, if this is my glucose here, then I draw an arrow going in this way, and then 2 arrows coming out, because 2 molecules of glycerol are produced that way, so that you can see that the glucose is going in, the glycerolase is coming out. And then because of that, hang on, let me get rid of my pen here. And then because of that, let me get back here, we now again have an equal amount of water going in as coming out. Remember, it's not that the water stops entirely, but that we're at equilibrium. So now the C. elegans is not dehydrated. And it can go about its life and have a really great time. So if you have any questions, feel free to put them on Schoology. I'm actually going to open up kind of a discussion on Schoology, and then right now we are working on getting access to Microsoft Teams so that we can have kind of open office hours for you to ask where people can show up and then you can hear other people's questions. But I want to really use Schoology right now instead of just emailing questions so people can see what other questions people are asking. Have a good day.